Hello everybody, and with this video we continue to discuss adaptive immunity, and in this part we will talk about humoral immune response, more about B lymphocytes and antibodies. Okay, so adaptive immunity, it's an experience of the life of each individual, of each person, and it can be humoral and cell-mediated. Cell-mediated immunity associated with T killer, T cytotoxic lymphocyte, CD8 cell, and humoral immunity is associated with antibody production. Humoral immunity refers to mechanisms of the adaptive immunity defense that are mediated by antibodies secreted by B lymphocytes or B cells. Okay, let's discuss B cell. B cells are formed from multipotent hematopoietic stem cells in the bone marrow, as all cell of immune system. B cell follow a pathway through lymphoid stem cell and lymphoblast. B cell continue to mature in the bone marrow to become B cell. As T cell, B cell also have a step of maturation, have a step of selection to not be autoreactive. So in B cell maturation, there are two steps: positive selection and negative selection. Positive selection for B cell with normal function, the antigen binding receptor, and negative selection is then used to be eliminated cell-reacting B cell. Immature B cells that pass through the selection in the bone marrow and travel to the spleen for their final stage of maturation. There they become mature B cell, mature B cell that have not yet been activated. Okay, look at this picture. You can see that this is polypotential stem cell, and this is cell which will be B lymphocyte, and this is B lymphocyte with its BCR receptor. BCR receptor, B cell receptor, the maturation of B cell involves becoming immunocompetent, meaning that it can recognize and bind to a specific molecule or antigen. This recognition results from the presence of highly specific receptor on the surface of B cell. On B cell, this receptor contains antibodies which are responsible for antigen binding. BCR, actually, it's a immunoglobulin D, antibody class D. An antibody is specific for one particular antigen. Typically, it will not bind to anything else. Difference between BCR and TCR. TCR is a receptor of the T cell. BCR is a receptor of B cell. TCR can only interact with antigen epitopes that are presented with the antigen binding left or MHC1 or MHC2. BCR do not require antigen presentation with MHC. They can interact with epitope on free antigen or with the epitope displayed on the surface of intact pathogens. TCRs only recognize protein epitopes. BCRs can recognize epitopes associated with the different molecular classes, proteins, polysaccharides, lipopolysaccharides. Each B cell initially produced has only one kind of antibody antigen receptor, which makes every B cell unique. It is the immense number of B cells in the body, each of which produce a unique antibody that allows the immune system to detect such a wide variety of pathogenic antigens. So here we can see B lymphocyte and its BCR receptor. Actually, BCR receptor it's a immune globulin which have an antigen binding site to catch new antigen, to recognize the antigen. B cell activation of B cell by T-dependent antigen. When B cell encountered the antigen that bind to its receptor, the antigen molecule is brought into the cell by endocytosis, reappearing on the surface of the cell bound of the MHC class second molecule. When the process is completed, B cell is sensitized. The sensitized B cell must then encounter a specific kind of T helper before it is activated. This activation of T helper cell occurs when a dendritic cell presents an antigen on its MHC second molecule, allowing T cell to recognize and mature. The helper T cell binds to the antigen MHC class second complex, and it is induced to release cytokines that induce the B cell to divide rapidly, making thousands of identical clonal cells. This daughter cell become either plasma cell or memory B cell. So here we can see the process of maturation of B cell, and then they became the plasma cell. Plasma cell will produce the so they make a clonus of the plasma cell, and plasma cell will produce antibodies, and also B cell will become the memory cell, which will take part in the secondary immune response. The memory B cell, a later encounter with the antigen caused by a reinfection by the same bacteria or viruses, will result in memory B cell divided into a new population of plasma cell. The plasma cell produce and secrete antibody molecules. Cell preparation in the antibody. So this is a very difficult process. It's a really difficult process, and I will try to explain it clear about. Okay, so the beginning of this process, it's antigen presentation. Antigen presented cell will activate T helper. T helper recognize a new epitope from its TCR because they can touch the MHC second from APC. So TCR became T helper second. And then T helper second send the signal to the B cell. And B cell also start to catch a new antigen. So look here, look at this process. Look at this process, so this is a T helper which was activated after interaction with APC. T helper will interact with B cell. Will send the signal to B cells that we have a new pathogen. You need to recognize it, and B cell catch the antigen by its BCR receptor, destroy it, catch only its peptide epitope, and these epitopes go inside the B cell, and then this epitope of so antigen determinant from new pathogen be uh, will be presented in MHC second receptor, and CD4 activated CD4 after activation with APC will check and send the signal, yes, this is correct. And then after T helper submission, B cell start to be activated, and activated B cells start to produce plasma cell, and plasma cell will produce antibodies. So phase of immune response. The first step is the antigen recognition. The second step is the activation of B lymphocytes. And the third step is the effector phase when antibody is produced. So the first step is the recognition new antigen by B cell by T cell, activation of T helper, activation of B cell, proliferation B cell to plasma cell, and effector phase is producing an antibody. And the third step, it's a memory step when memory cells are created. Memory cell from B cell and from T helper. And direct interaction of B cell and T cell involves co-stimulator molecules. So you know that T helpers, they interact with antigen presented cell. And T helper, they proliferate into T helper 1, T helper 2. T helper 1, they most take part in the cellular immunotransfer in activation of CD8 cytotoxic T killer cell. But T helper 2, they take part in activation of B cell because they interact with its MHC second receptor, and they when B cell catch new pathogen and they submit that it's a correct pathogen, and then also they stimulate proliferation of B cell into plasma cell by production of different cytotoxins like interleukins, so 4, 5, 6, 10, 13. And cytokine secretion is important in B cell proliferation and differentiation. And actually, I need to say that cytokines, they like messenger molecules by which all cell of immune system interact with each other. B cell activation also can be T independent without T cell. A small number of antigen can be activated B cell without MHC class second restricted T helper and are referred to the T independent antigen. T independent antigen, T1 antigen are predominantly bacterial cell wall components like lipopolysaccharide. T independent 2 antigen are predominantly large polysaccharide molecules with repeating antigen determinants like dystran, polymeric bacteria, flagellin, and polymerase viruses. T-independent activation, BCR interact with T-independent antigen. T-independent antigens have repetitive epitope units with their structure, and this repetition allow for the cross-linkage of multiple BCR. Once BCR is activated and undergoes clonal proliferation and daughter cell differentiated into the plasma cell. Plasma cells are antibody factories. After differentiation, the surface BCR disappear and the plasma cells secrete pentameric immunoglobulin M molecules that have the same antigen specific of the BCR. The T cell independent response is short-lived and does not result in production of the memory B cell. This is very important. So T independent B cell activation occurs without T helper. So T helpers, they don't take part in this process. Just activation of B cell by interaction of the T independent antigen. And B cell after this activation start to proliferate to the into the plasma cell, and plasma cells start to produce immune globulins M. But but this process is undergoes without production of B memory cells, so without memory. Okay, so we just know that antibodies, it's a main part of the humoral adaptive immune response. So what is antibodies? Antibodies are glycoproteins that are present in both the blood and tissue fluids. The antigens of humoral adaptive immunity and agents of humoral adaptive immunity, antigens is produced by plasma cell after stimulation by an antigen. Antibodies can bind pathogen and mark them from destruction by phagocytosis. phagocytes before they are able to infect cells. Structure of antibody. The basic structure of antibody monomer consists of four protein chains held together by disulfide bonds. And the two largest chains are called the heavy chains, and two smaller chains are called the light chains. Joined together, they have the heavy and light chains form a basic E-shaped structure. Two arms of the E-shaped antibody molecules are the Fab region fragment of antigen binding. The Fab region is a variable region. The Fab region is capable of bind of the epitope. The constant region of the antibody molecules include the trunk of the E and lower portion of each arm of the E. The trunk of the E is also called the FC region for fragment of crystallization and is a site of complement factor binding and binding of phagocytic cells during antibody-mediated opsonization. So here we can see the antibody. Antibody have a heavy chain and light chain, and two chains are bound together by disulfide bonds, and with light chain also by disulfide bonds. And this is a Fab part, so antigen binding fragment to interact with antigen. And this is a fragment of the crystallization FC region to interact with complement, to interact with phagocytic cell in the process of phagocytosis and opsonization. So in the human body, there are 5 classes of antibodies: immunoglobulin G, M, A, D, and E. Immunoglobulin G, it's a large part of immunoglobulin. So in our bodies, there are more immunoglobulin G than other antibodies, and it has a 2 antigen binding sites. So they looks like that structure. And so it is about 80% from all antibodies in our organism, and they are the smallest immunoglobulins. That's why they cross placenta, and also they are the second immunoglobulins which are produced in immune process. Okay, immune globulins M, they are the largest immune globulins, and because they have 10 antigen binding fragments, and they not live too long life. That's why the amount of immunoglobulin M in the serum is about only 6%, and they are very big. That's why they can't cross placenta, and actually only immunoglobulins G can cross placenta. And they are the first immunoglobulins which are produced in the immune answer. That's why if we catch immunoglobulin M in the human plasma, that means that this person in the stage of acute infection. Immune globulins M, it's a marker, it's a sign of acute infection. Immune globulins A, they are dimer, so this means that have four antigen binding site, and they present in the mucous tissue, and they make a defense on the mucous tissue. Immune globulin D, it's a BCR receptor. So they present on the surface of B cell, and their function is to catch antigen and make its presentation. Immune globulin E, it's a monomer, and they take part they activate basophils and mast cell, and they are active against parasites like protozoan and nematodes, and also immunoglobulin E take part in allergic reaction. Okay, so we can see more about each of these uh, immunoglobulins. Okay, effects of antibodies. What, how antibody can take part in the protection against infection? They can neutralize, they can't destroy bacteria, but they can bind with their adhesins and destroy the process of bacterial adherence. So they can stop the process of bacterial adherence. Also, they are opsonins, so they take part in opsonization. If you remember, opsonization, it's a help in the process of phagocytosis when phagocytic cell meet a bacteria, meet a pathogen. Phagocytic cell need to recognize that it is not self, that it is something foreign, and uh, this recognition is occur with the touching of bacterial antigen. But if bacteria are marked with antibodies, phagocytic cell understands that it's something foreign faster. So the opsonization is the help from phagocytic cell in the process of recognition and engulfing. And complement activation, you know, the complex antigen antibody activate complement by a classic pathway, and complement after activation will destroy, will kill pathogen, will kill bacteria. Okay, neutralization involves the binding of specific antibody to antigen but found in bacteria, viruses, and toxins, preventing them from attaching to the target cell. Here about opsonization and also antibody-dependent cell-mediated cytotoxicity. Okay, antibodies bind to a large pathogenic cell that is too big for phagocytosis and then bind to Fc receptor of the membrane of natural killer cell. This interaction brings the natural killer cell into close proximity where it can kill the pathogen through release of lethal extracellular cytotoxins. Cell-mediated immunity, cell-mediated cytotoxicity is an essential defense against intracellular pathogens, including viruses, some bacteria, and some parasites. CTL cells and natural killers are the lymphoid factors of cytotoxicity. Okay, cytotoxic cell activation. Cytotoxic cell, also referred to cytotoxic lymphocytes or CTLs, CD8 cell or T killer, are activated by APC in a three-step process similar to the helper T cell. The activation of cytotoxic T cell involves recognition of an antigen-presenting cell with MHCN1 and interaction of CD8 with the receptor complex. After the successful co-recognition of foreign epitopes, the self-antigen, the production of cytokines by APC, and the cytotoxic T cell activated clonal proliferation and differentiation. Activated cytotoxic T cell can differentiate into defector cytotoxic T cell, then target pathogen for destruction of memory cell that are ready to respond to subsequent exposure. Proliferation and differentiation of cytotoxic T cell is stimulated by cytokines secreted from T1 cell activated by the same foreign epitope. The co-stimulation that comes from this T1 cell is provided by secreted cytokines. So, in the cell-mediated immune response, also it is necessary to be to activate CD4 and CD8 cells. CD4 it's a T helper. CD8 it's a T killer. So when antigen presented cell catch a new pathogen, it will destroy it and put its epitope to the MHC second receptor and will go to the T helper CD4 cell and will show it to T helper. T helper recognize a new pathogen with each TCL receptor and activate and became to be activated, and then T helper proliferate to T helper 1 and activated T helper 1 sent to cytokines to the CD8 cell. CD8 cell start to recognize antigen by MHC receptor of antigen presented cell, because antigen presented cell, it's a dendritic cell, it's a macrophage, and of course they have not only MHC second but also MHC1 receptor. And with MHC1 receptor, antigen presented cell shows a new antigen to the cytotoxic lymphocyte, to the T killer, and T killer are activated by this stimulation. So this is co-activation. So CD4 cells, they send the signal to the CD8 cells that they need to recognize MHC1 receptor. And by this way, CD8 are activated and start to proliferate to the clones, and this clones will go to the war and will find a new start to find a new antigen in the MHC1 receptor of the infected cell. So look at this scheme. This is scheme of TCR activation of the this is the stage of T cellular immune answer. So the same stage as it was in humoral immune answer. So antigen recognition by T cell and by CD8 cell, and then process of lymphocyte activation of T helper, clonal explanation to T helper second, and they also send the signal to the CD8 cells. CD8 are activated and start to proliferate into the clones of activated CD8 cell effector T helper cell. And from T cell, memory cell will be occurred, and effector CD4 cell, which activate macrophages, B cell, other cell inflammation. But effector CD8 cell, they will kill the target cell, and also memory CD8 cell. Cytotoxic T cells serve as the effector cell of cellular immunity, recognizing and kill cell infected with intercellular pathogens through a mechanism very similar to that of natural killers. However, whereas natural killer cell recognize non-specific signal of the cell stress of abnormality, cytotoxic T cell recognize infected cells through antigen presentation of pathogen-specific epitopes associated with MHC1. So natural killer, as you remember, they just check MHC1 receptor. Is it normal or abnormal? But cytotoxic T cell T killer, they find in the MHC1 the epitope of antigen which with which T killer was activated, by which T killer was activated. So you know that in the specific immune answer, in adaptive immune answer, activation of the T killer, activation of B cell are associated with one antigen, one epitope. So T killer, they will uh, find MHC, and they will need to find antigen in MHC1. So this is specific reaction, but natural killers, they are non-specific. They are part of non-specific immune response. Once an infected cell is recognized, the TCR of the cytotoxic T cell binds to the epitope and releases perforin and granzymes that destroys the infected cell. Perforin is a protein that creates a spore in the target cell, and granzymes are proteases that enter to the pores and induce apoptosis. This mechanism of programmed cell death is a controlled and efficient means of destroying and removing infected cells without releasing pathogen inside to the infected neighboring cell, as might occur the infected cell were stabilized. Okay, primary and secondary immune response. Primary is the reaction of the immune system when it contacts in an antigen for the first time, and secondary response is the reaction of the immune system when it contacts with an antigen for the second and subsequent times. Um, During the primary response, some of the cloned B cell are differentiated into memory B cell and T cell into memory T cell, programmed to the responses to subsequent exposure. And the secondary response develop more quickly and produce high and more sustained levels of antibody with high affinity for specific antigen. So look at this picture. Here we can see primary antibody response. So when immune system meet with antigen in first time, and you should notice that in the immune response is slowly, and maximal the higher level of antibody will be only on the 7 day or more, 20 day, like that days, and it is not so strong, because look here, this is a secondary immune response when immune system meet this antigen in the second or other time, or any other time, uh, and in the primary immune cell, it is necessary to activate B cell, to activate T cell, but in the secondary antibody, in the secondary response, it's just enough to have a memory cell, and memory cells start the immune response, and the higher level of the antibody will be on the third day of the exposure, and the level of antibody will be bigger than it will be in the primary response. So that's why we can say that secondary response is faster and stronger than primary immune response. Okay.